The CRISPR-Cas9 technology is used in cutting DNA at a precise location. It allows scientists to edit DNA and possibly cure genetic disease.
In this video, we will focus on three topics. How does CRISPR-Cas9 function, CRISPR-Cas9 application, as well as the controversy and safety concern. Firstly, let's have a look at how does CRISPR-Cas9 function. The CRISPR system is naturally used by bacteria to protect themselves from virus infection. When a foreign virus DNA plasmid invades the bacteria, it will be integrated into the CRISPR locus of bacterial chromosome as spacers. The CRISPR locus is then constitutively transcribed into pre-cRNA. The constitutively expressed trcRNA will base pair with a CRISPR repeat sequence on pre-cRNA. RNA3, CS1, and other protein will modify the pre-cRNA and trcRNA duplex to form a guide RNA. The guide RNA binds to Cas9, an endonuclease that cut DNA, and form a complex. The guide RNA Cas9 complex will then be activated. It binds to the invading DNA and reads through the DNA. When the guide RNA detects the virus DNA, the Cas9 will cut the virus DNA and disable the virus. Let's have a close look at how does CRISPR-Cas9 complex function. Firstly, the Cas9 protein needs to locate and bind to a DNA sequence called PAM. After Cas9 and PAM binding, the guide RNA will unwind part of the DNA double helix. The guide RNA will recognize and base pair with the virus DNA. Each of the two domains of the Cas9 will make a nick and lead to the break of virus DNA. The virus DNA will attempt to repair itself. However, the repairment is error-prone and often causes mutation. Talking about CRISPR-Cas9 application, scientists have found that the Cas9 and guide RNA complex can be used to cut almost all DNA sequence by editing the guide RNA sequence. The cell will try to repair the broken DNA after DNA cutting. However, this generally leads to mutation. Research has proved that the mutation sequence can be replaced by a different DNA copy by simply adding the copy into the cell.
Recent study indicates that CRISPR technology can be used in therapeutic treatment of HIV. HIV-1 used cell receptor CCR5 as a mode of viral entry in CD4+ and CD8+ T cells. The discovery of a naturally occurring mutation of delta 32 on chromosome 3 found that homozygous individuals are highly resistant to HIV infection. Thus, the ablation of CCR5 using CRISPR technology could be a key strategy to treat HIV infections. CD34+ is a progenitor for CD4+ and CD8+ cells. CD4 and CD8+ cells would retain delta 32 CCR5 mutation in the edited CCR gene when CD34+ differentiates.
The procedure for the CCR5 knockout in the human hematopoietic stem cells is shown as follows. In this video, we will emphasize the mechanism for CCR5 knockout and how it provides resistance to HIV as a result.
Phase 1 of CRISPR knockout involves the delivery of SP-Cas9, a recombinant isophenogene's Cas9 nuclease, and specific single guide RNAs. Cas9 was cloned with a pCNV promoter into simian viruses for delivery into human CD34 cells derived from human fetal liver. Single guide RNAs target the locus from the beginning of the first exon to the delta 3 mutation site in the human CCR5 gene. Cas9 then nicked the targeted site. A frame shift mutation CCR5 using this technique would inactivate the CCR5 gene, and CD34+ would not express the chemokine receptor. The results showed CRISPR technology is successful in CCR5 gene knockout.
There are some controversy and safety concern for CRISPR-Cas9 in gene editing. One of the challenges is the off-target effect. where Cas9 cuts the target gene with 3 to 5 base pairs possible mismatch. The PAM sequence is 2 to 6 nucleotides adjacent to target gene. Off-target cleavage could occur and lead to malignancy diseases. The concentration of Cas9 nuclease and Cas9 gene expression time are important in minimizing off-target activity. Also, a vector without any toxicity is required to deliver the gene of Cas9 nuclease and guide RNA in gene editing. Moreover, there is an ethical concern in genome editing, like manipulating human embryonic genome, as these alterations would be permanent. The long-term consequences are indefinite. Therefore, using CRISPR-Cas9 to alter human embryo is currently restricted.